Hello, good morning to all of you. Welcome to IMMB International Webinar Series. My name is Mohammed Hakim and I am a chairperson for today. The title of our talk for today is Macro and Microenvironment Considerations of Designing Future Foods, that will be presented by Dr. CHING Jianhong. A bit introduction for Dr. CHING Jianhong. Immediately after Dr. Ching completed his PhD at the Department of Pharmacy, National University of Singapore, he trained at Duke University and performed a technical transfer to build Singapore's first fee-for-service metabolomics facility at Duke-NUS. The facility headed by Dr. Ching has since taken part in numerous large-scale clinical screening on metabolomics in collaboration with most major hospitals in Singapore. Dr. Ching is appointed as joint scientist for the KK Women's and Children's Hospital and also invited as a scientific advisory board member of MD, Southeast Asia First Precision Gut Microbiome Company, where he applies metabolomic techniques on gut microbiome projects. Dr. Ching enjoys applying his metabolomic skills in different fields and has been invited to advise on Singapore's novel food regulatory framework at the Singapore's Food Agency's Novel Food Expert Working Group. Dr. Ching is also the Vice President of the Singapore Society of Mass Spectrophotometry, SSMS, and the Strategy Engagement Manager of the Association of Official Analytical Chemists, or AOAC, Southeast Asia sections. That's all. Okay, we can proceed with the agenda of the day. Please, Doctor.
Thank you. Yeah, thanks for the very kind introduction. And let me reiterate again that I hope that this talk would be more interactive. So if you have questions, feel free to type on the chat box anytime. Yeah, so I'll try to address them as much as possible. Let me share screen. Let me know if you're looking at the correct screen. Okay, are you looking at the correct screen right now? This is the slides and not the notes. Is that right? Yes, thank you. Right, so good morning again, everyone. My talk today is on the macro and micro environment considerations of designing future foods. And in this talk, other than telling you the story of sciences behind alternative proteins, I hope to also impress upon you the importance of multidisciplinary approaches in science. Besides food science, this topic comprises of science from environmental studies, analytical chemistry, molecular biology, and even microbiology. I'm Jianhong, an assistant professor from Duke-NUS Medical School. Duke-NUS is one of the 3 medical schools in Singapore. But Duke-NUS only takes in graduate students. We have 5 signature research programs: cancer, stem cell, neuroscience, emerging infectious diseases, health services, and cardiovascular and metabolic disorders program. I'm an analytical chemist by training and biologist by training, which means I'm a specialist at measuring compounds in the biological system, also known as metabolites. Because of my expertise, I'm recruited to the CVMD program, or the Cardiovascular Metabolic Disorders program, where I help the researchers in the school measure and analyze the metabolites of their sample. The interesting thing about this is that a few years ago, when alternative proteins became important as a tool against climate change, novel food became a trend in Singapore also. And this is because Singapore has a very small land area which cannot be partitioned for agriculture. So Singapore began to seriously look into producing novel foods such as meats in the lab. And this process is actually nothing new in the lab, which is also known as cell culture. But to use the cells for experiments and to eat them are totally different methods altogether. Because if you have done cell culture before yourself, You'll be adding things like antibiotics, growth hormones, and all kinds of drug and drug-like compounds to induce differentiation. Oh, there's a message here. Oh, yeah. Feel free to chime in anytime for questions. Thank you very much. Yeah. So my interest in food safety also led me to become the public engagement manager of the AOACC, a non-profit organization. This is a section of the AOAC International at Southeast Asia, and has many student-related activities, which I'll tell you briefly later. And finally, I want to highlight that I am also engaged as the advisory board member of IMMB, a fecal bank founded in Singapore, which also has links to Kuala Lumpur's Monash University's Microbiome Center. The reason I will tell you in the second half of the talk. To start the lecture, we first talk about the macro environment, to the environment that we know very well, and this is the environment that we live in, the Earth. We know that the Earth is undergoing rapid climate change now, and this is one of the main reasons for driving alternative protein production. Our global food system currently causes 34% of global greenhouse gas emissions, and it's believed that plant-based meat uses up to 99% less land than conventional beef, and cultivated meat could use 95% less land than conventional beef also. Other than being beneficial for the forests by minimizing deforestation, there are also marine and aquatic benefits, since alternative proteins can potentially reduce fishing, so it reduces habitat destruction, and also reducing agriculture land use and inputs means improvement of the water quality. Improving the water quality and reduction of ocean pollution and degradation. So here we look at the timeline of agriculture history. Some of the major events are that of the usage of the fertilizers in the early 1900s, the creation of the genetic modified foods during the 1970s. Then we currently have alternative proteins in the 2020s. Sorry, there's a typo here, 2020s. So we are actually living in a turning point of agriculture history. Impossible Meats, plant-based meat, was just launched in 2021, and Eat Just, cultured chicken meat, was just authorized for sale last year, 2022, in Singapore. So when we talk about alternative proteins, we tend to use the term alternative proteins and novel food very interchangeably. However, from the regulatory point of view, we want to only create new regulations for new kinds of foods which has never been eaten before, and not for something that has a history of consumption, but just that they are not very frequently being eaten. Take insects, for example. While insects are considered a new kind of food to the Western world, in parts of Asia and Africa, insects have been consumed since human history, and is hardly considered novel. Hence, under the Singapore Food Agency regulation, novel food and alternative proteins do overlap, but have their own definition. For novel foods, they are food ingredients with less than 20 years of human consumption history, and this also includes traditional foods that are produced through advances in biotech. And alternative proteins are alternate, of course, to what is mainly being consumed. For example, poultry, beef, and other livestock. And examples of alternative proteins are plant-derived proteins, microproteins, algae proteins like spirulina. I think many of you have maybe seen or consumed this before. Cell-based protein or cultivated meats, and insect protein. The novel foods like cultivated meats and alternative proteins are plant-based meat, and these are mainly to satisfy meat eaters. So we are trying to produce a food source that resembles their habitual diet. And some requirements for alternative proteins include it being cheap enough to buy, of course, and taste the same or better than the real meat. So people will be willing to try them. And other than the usual plants and animals, for alternative protein, we also have to consider what are the more unusual food sources like insects and microorganisms. Insects are already regularly consumed in certain parts of the world like Asia and Africa, and are widely known for their high protein content. Microorganisms are also getting more popular now, although it has been used many years ago for the fermentation of foods like kimchi, yogurt. They're also grown and used for protein directly, like Marmite, yeah, and also in medicine production like insulin. So basically, in the latest technology, what we call precision fermentation, the genes which are the blueprint to construct a protein is inserted to a microorganism to enable it to synthesize proteins with amino acid sequences identical to those derived from animals. So these proteins are then separated from the microorganisms and used as an ingredient in other foods. The brand, the ice cream brand Perfect Day, here makes an ice cream with the protein produced by the microorganisms, while every company creates eggs via a fermentation process. So what are the benefits of each kind of alternative proteins? For plant-based meats that have they have a very huge potential in bringing down carbon emissions, more than 90% in land use, more than 80% to 90% in reducing greenhouse gas emissions, and same for water usage. However, cultivated meats are more controversial still. According to the MIT Technology Review, there can be two kinds of scenarios. A food grade process to process the meat results in an equivalent of 10 to 75 kilograms of carbon dioxide emissions, lower than the global average emissions from beef. But in the biopharmaceutical-like process, cultivated meats lead to significantly more emissions than beef production today, between 250 to 1,000 kilograms of carbon dioxide equivalent for every kilogram of beef, depending on the specific scenario. So yesterday I was in the Novel Food Regulators Forum, and they were discussing, should you be using the food process or the biopharmaceutical process? It's still under debate right now, because right now we don't even know how to go about regulating or how to go about testing. So we tend to want to be more careful in all these things. So still discussing, and really the carbon emissions are something that is very serious, and we need to take into consideration also. So for insects, the climate advantage is more obvious. Okay, they use up to 50 to 90% less land per kilogram protein produced, 40 to 80% less feed per kilogram edible weight, and produces 1,000 to 2,700 grams less greenhouse gas emissions per kilogram mass of than the conventional livestock. So we should eat more cockroaches and so on. So it is believed that the market for alternative proteins will grow tremendously. And in Singapore, a $60 million fund had been allocated for such kinds of work and research. And if I'm not wrong, this field is also getting very popular in Malaysia too. I'm seeing many novel food conferences in Malaysia this and last year, and just last week there was the 17th ASEAN Food Conference held at Kuching, which many of my colleagues attended also. Of the various alternative proteins, the most difficult to regulate would be the cultivated meats. This is because unlike plant-based meats, which essentially are from plants, we have always been eating plants, the insect, algae and microorganisms which has a history of being used, okay, also, but the cultivated meats do not have a history of being used. So, and also there are many methods of producing them, and these methods are proprietary and difficult for regulators and scientists alike to understand. Some of the concerns include finding out whether the meat replacement has nutrients equivalent to that of the traditional meats. And it is reported that the bioavailability of certain nutrients are not as high as that in the traditional meats. Also, cultured meats might be lacking in certain metals or nutrients like iron, because when we eat meat, the hemoglobin or myoglobin contains iron. That's how we get iron from food. But simply, this iron can be missing, absent from the cell cultures, because right now they are using very specific cell cultures like the stem cells, the muscle, the fat cells, and so on, which may or may not have existence of hemoglobin or myoglobin. Maybe muscle cells might have. And those who do cell cultures know that there are many components that you have to add into the cell cultures, like growth hormones, antibiotics, and all these require a system for regulation. What is the purity that's needed, and so on. Yeah. After regulating the externally added components, we also need a system to monitor if the cells are changing by themselves, by detecting mutations, etc. And mutations might lead to chances where toxic components are created, or there's an upregulation of proteins that causes allergenicity. So this is where I come into the food scene, although my main research is on metabolic disease. So my role as an analytical chemist and a member of the Singapore Food Agency Novel Food Expert Working Group is to advise them on the safety of such foods. Unlike traditional foods where the safety checks are clearly defined, cultured meats are grown using technology that's unfamiliar to the regulators. And there's someone like me who understands cell culture and compound detection. What I tell them is that they can look at the chemicals of the real meat here, and compare to those of the cultured meat here. Yeah? And those that are from the real meat can be assumed to be safe, and what is different from the cultured meat must be further investigated. And this is essentially how biomarkers are discovered from biomedical research in diseases. Other than a simple Venn diagram, another technique is to model using PCA plots, or principal component analysis plots, shown here. We do this traditionally for stratifying patients, for example, cancer stages. In biomedical research, when we want to look for biomarkers in the disease, we'll collect specimens from the patients at different stages of the disease, and after running them on a mass spectrometer, the data is reduced in dimensions mathematically into a PCA plot. And the different stages will cluster together like this in this diagram, like stage 1 cancer, stage 2, stage 3, stage 4, and so on. So this is essentially called modeling. So when in the future, a patient with unknown disease stage needs to be stratified, we would run the sample from him or her, then match it to this database. Using the same concept, we can run several batches of cell culture meat to model what belongs to an acceptable quality control. If a cell culture batch falls beyond any of the groups that we have modeled before, then this warrants further research and investigation into the safety of the culture. What I have mentioned here are short-term safety issues that we have to screen for. But there are, of course, long-term safety issues that we have to be aware to. One issue I brought up to the committee was about the cleanliness of the meat. Cell cultured meats are very clean, as stated here. They have extremely low microbiological content. And you might think that if they're cleaner, very clean, isn't it better? Then you won't get diarrhea and so on. Huh? So what's the problem? You have to understand that not having your usual microbes in your food can actually have detrimental effects to your health. And this leads to the next part of the story. After talking about the macro environmental effects on health, we will now talk about the micro environment within our body. I liken the human body to a universe itself, having an internal system that acts like a planet Earth in response to an outside environment, the universe that's not within our control. In traditional Chinese medicine, in Ayurveda, which is Indian medicine, and even ancient Greek medicine, the principles compares the human body to the universe, philosophically and mystically involving the balance of elements of nature within the body. Modern science now agrees that we are not just one person, one being, but we are a universe housing 39 trillion other beings. A reference man who is 70 kilos and 20 to 30 years old, 1.7 meters tall, contains on an average of 30 trillion human cells and 39 trillion bacteria. So it is common knowledge that future food stems from the need to protect the environment. And this next story, which I'm going to tell you, furthers the concept of the environment within our body. But within our body is actually not an accurate term, and you shall see why here. This is a diagram showing the development of an embryo. For the simpler and lower organisms, which is shown on top here, after the cells divide, this yellow part of the ball of cells eventually becomes the blastophore, which becomes the mouth of the organism. On the other hand, complex animals like us, okay, the same blastophore becomes the anus, okay, becomes the anus and linking all the way to the mouth. And what is in the middle becomes the gut. So you would notice that the human gut actually is never formed from within, and it's actually formed externally because this is the ball of cells, and then you just form a cleavage like that. So whatever that is mouth to anus is actually outside, all the while it has been outside our body. So since this is an external environment, this external environment will of course be influenced by the macro or outside environment. So you might know that the outside environment is not sterile, okay, and this comes the question of many problems, how to control and what does this impact? How does this impact the health? So when the mother gives birth to a baby, the baby actually inherits the beneficial bacteria that is similar to that in the birth canal. Whereas those babies given birth by cesarean, they inherit that from the surgical room. There are studies that link the increased observations of allergies to C-section babies due to the interrupted transmission of the maternal microbiome to the infant. Additionally, antibiotic exposure during the early years of a human can cause lasting damage to the body by disrupting beneficial gut microbiome. And this leads to increased risk of metabolic diseases. This gives evidence the importance of gut microbiome during to the health of humans. In our adulthood, it is widely known that the gut microbiome you have is heavily affected by the food that you eat, which is influenced by the climate which you stay in, your race, your culture and religion, depicting your eating habits. So someone consuming a typical Western diet that's heavy on carbohydrates and fat will have very different gut microbiome from someone who is vegetarian. And this person will also be very different from someone who takes a diet heavy on spices like the Indian diet. It's clear that different geographical regions have your own unique set of microbiome. So you will notice that when you travel overseas, sometimes you get diarrhea when you're eating exactly the same thing as what the locals are eating, but they're perfectly fine. This is not because what you have eaten something dirty or something, because everybody is eating the same thing. But this is due to a change in your gut microbiome, and then your immune system is acting on the new microbiome. And this set of microbiome which you have is also affected by internal factors such as your genetics, your mental health, and also exercise. So there are some people who have a lack of lactobacillus in the body, and those with neurological issues such as autism is known also to have a different set of microbiome. While I'm advising the Singapore Food Agency on food, I'm also advising a poop company, fecal company called IMMB, or the Asian Microbiome Library. IMMB is the first Asian fecal bank, and this is a news article featuring it. Here they are describing the health effects of the gut microbiome and the diseases that they influence. That's literally from the head to the toe. Yeah, so from the neurological or brain-related kind of disease all the way to diabetes and arthritis and so on. In IMMB, what they do is that they collect good poop from healthy people, those gym instructors and aunties who does yoga and so on, very healthy, and they only eat vegetarian food. They take the poop, okay, and then they give them to patients after they screen and they know what could possibly be lacking in their microbiome diversity. The doctors in the company had performed what you call FMT, or fecal microbiota transplant, for patients with irritable bowel disease, and ultimately the aim is to purify the bacteria and feed them to patients to cure diseases. The rationale behind fecal microbiota transplant stems from research such as this, where they took feces from genetically identical twins, but one is obese and one is slim, and transplanted them to genetically identical mice. All the mice are the same, okay? And the mice who received the feces from the obese twin became obese, okay? And the mice that received the slim twins feces remain slim. And what's interesting is that after this slim mice remain slim, they took the mouse, the slim mouse feces, and then they put it into the obese mouse. The obese mouse became thin. Okay, this highlights the profound effect of gut microbiome has on our body and metabolism. Researchers like me are still trying to find out the mechanisms by which these bacteria exerts its influence on our body. And one thing which we understand is that these bacteria ferment the food in your tummy, and then they produce chemicals. The carbohydrates become short-chain fatty acids, and then and other gases, and the proteins become things like ammonia, branched-chain fatty acids, phenol, amine, sulfides, etc. What do they do? I'm not too sure. Still trying to research. There are some work being done. For example, some of the compounds called the short-chain fatty acids. Okay, the short-chain fatty acids are very, very smelly. They smell like vomit. That's why your vomit, these things are in the gut. That's why your vomit smells like vomit, because they contain a lot of short-chain fatty acids like butyric acid. Okay, so now you know why vomit smells like vomit, because they have a lot of short-chain fatty acids in the gut. So these short-chain fatty acids bind to receptors on the cell walls of various organs and give a wide range of effects, like increasing satiety, increasing insulin sensitivity, decreasing plasma glucose levels, which is good, increasing fatty acid oxidation so that you burn more fat, and so on and so forth. A lot of different effects. It is until now still very difficult to understand how different gut microbiome affects the human health. So in this paper, a very, very recent paper published just last month or something, just 1 or 2 months ago. Okay, they try to understand how the different gut microbiome affect the human health. They find that the microbiota reprograms intestinal lipid metabolism by repressing the expression of long non-coding RNA, or what you call the cRNA. This RNA called the SNHG9, small nuclear RNA host G9. And this SNHG9 suppresses PPA gamma, a central regulator of lipid metabolism. When the gut microbiota is not present, or when there's forced expression of SNHG9, so you purposely increase the expression of SNHG9, that's impaired lipid absorption and reduce body fat, and thus this protects against diet-induced obesity. So in the mice where they remove all the bacteria, they stay slim, whereas the animals with intact microbiota have lower levels of the long-chain LNC-RNA and are fatter. Okay, so let me ask you a question here. Is microbiome good or bad? Okay, some microbiome helps to store fat. Is this beneficial for health? Okay, so it really depends on the environment. In an environment where food is scarce, the gut microbiome that helps you to store fat is good. But when food is aplenty, then this is bad because then you eat a little bit and then you keep storing fat. So there are some microbiome that helps you to store fat. There are some microbiome that helps to burn more fat. So it really depends on the environment. Okay. By evolution, we are born to have gut microbiome that helps us to store more fat, because in prehistoric days when we do not have regular food intake, so storing more food in the body or storing more energy in the body is better than not storing. But right now, this is opposite. Evolution is targeting people who are slimmer, healthier, because the fat people die of cardiovascular disease more easily. So it really depends on the situation. And other than metabolically changing changing the metabolics and the disease, can also act on the brain and influence autism, moods, Alzheimer's disease, Parkinson's, and sclerosis. And there are reports on trying to alter gut microbiome for people with autism to treat the symptoms. But what was observed was that the people with autism has some kind of gastrointestinal disorder. So they think that it could be a problem with the gut microbiome. Success of the treatment has so far been anecdotal. So this warrants further research to see whether this actually works on autism or not. And more on the psychology, recent research also shows that certain gut microbiome can make you want to exercise more. Okay, so what was found was that the gut microbiome produced FAAs, which are fatty acid amides in the gut, and this stimulates sensory neurons. And signals from these sensory neurons leads to increased dopamine levels in the striatum during exercise, and dopamine in turn enhances the desire for exercise. All these are discovered on mice, but if these are confirmed in humans, and this could suggest strategies to help to encourage people to exercise. So people, friends who are here who are lazy to exercise, maybe you should look out for friends who like to exercise, and then not to get their poop and then do FMT on yourself, but to ask them what they eat, so that you can know how to maintain and develop this exercise microbiome. Yeah. Well, the example cited probably involves more long-term changes and influence the gut microbiome through the diet and environmental changes, eventually causing changes in metabolism and disease to take place. There are also examples in food that can cause acute changes that causes direct impacts on health. Recent studies shows that exposure to triclosan or TCS and triclocarban TCC at human exposure relevant doses increases the severity of colitis and exacerbates colon tumorigenesis in mice. So this suggests that they can be risk factors of irritable bowel disease and associated diseases. TCS is a common ingredient of toothpaste, but has since been banned. And the gut toxicities of these compounds require the presence of gut microbiota, since they fail to induce a colonic inflammation in mice lacking the microbiota. Regarding the functional roles of the microbiota involved, the commensal microbes and specific microbial beta galacturonidase enzymes mediate colonic metabolism of TCS, leading to metabolic reactivation of TCS in the colon and contributing to its subsequent gut toxicity. So actually, these compounds are cleared from the body already, but when the gut microbiome is there, they will reactivate the compound, and then it will cause more gut toxicity. Overall, these results support that these commonly used compounds could be environmental risk factors for IBD and associated diseases through gut microbiota influence. In a more positive sense, the gut microbiome can also do good things to your food. Stevia, stevia, you find it in Coke and so on, the Coke Zero. Stevia is a zero-calorie sugar substitute and is recognized as safe by the FDA and EFSA, the European Food Safety Authority. Extracts from the stevia plant contains stevioglycosides, which are about 50 to 300 times sweetness of sugar. The metabolism of stevioglycosides is dependent on your gut microbiota, which breaks down the glycosides into stevio, then it's absorbed by the host. Otherwise, it cannot be absorbed by the host. So after the stevia enters the host cells and they can have anti-glycemic actions, antioxidant actions in the adipose tissue, vascular wall, and this reduces blood pressures, reduce hepatic steatosis, and stabilizes arteriosclerotic plaques. And it also help to reduce liver and kidney damage. So drink more Coke Zero. And the anti-inflammatory properties of steviosides are confirmed by inhibiting that they inhibit TNF-alpha cytokines like IL-1 beta, IL-6 synthesis, and inhibition of the NF-kappa B transcription factors. There's a question. Is stevia 0 calorie? Yes, because in your human metabolism, you take in 3 kinds of nutrients. You have fatty acids, you have proteins, amino acids, and then you have glucose. The human system can only burn glucose by glycolysis to make energy, and it cannot use stevia. Where also because stevia is 300 times sweeter than sugar. So that means you take in very little stevia for the same amount of sweetness. So even if it acts the same as glucose, it's still considered almost negligible. Yeah, hope I answered the question. So from the examples, the gut microbiome can be a double-edged sword. The good microbiome helps you to maintain a healthy metabolism, and the bad microbiome can lead to diseases. And even when there's good microbiota present, it can also cause issues where you consume certain chemicals that can interact negatively with the microbes. Coming back to my point about cultured meats having no bacteria present, this can be a bad thing in the long term, as such food will certainly cause changes in your gut bacterial system. When the food is sterile, the normal microflora is absent. Okay, 2 scenarios can arise from this. First, long-term and large intake of such sterile foods, which is the eventual intention, can slowly alter the microbiome diversity of our gut system. And second, if the normal microflora is absent in the food, then another non-common microflora might want to take over, since the protective effect of the usual microflora is absent. In this case, the growth of this unique microflora can pose a health hazard to our body system, depending on what they are. So many of you might think, okay, then you just supplement the food with probiotics, can? No? Okay, then research, right, have shown that probiotics are not exactly the best solution. Human and animal trial shows that many probiotics just do not have any effect on human health, as they cannot survive in the gut long enough. And that's why you have to keep drinking probiotic drinks like Yakult. And lucky, Yakult is nice to drink. If not, nobody wants to drink Yakult. You can so you can see here some of the and also you see this study here when they have different species of lactobacillus put into the gut, they actually die off pretty quickly, like 20-30 minutes. This one will die off. Some will remain there. Okay, some survive moderately, but basically many of them will just die off very quickly. Yeah, so you have to continuously eat them. And at the other spectrum, some of these probiotics, right, can even prevent normal flora from recurring after a bout of antibiotics. In this study, you can see that when at first everything is the same, when you take antibiotics, all the species of the bacteria die off. When you just leave them alone, you don't do anything to the gut, they will slowly increase back. When you give them a fecal microbiota transplant, you implant back the gut microbiome, then they quickly return back to normal. But when you give them probiotics after antibiotics, the probiotic species actually suppress your normal species. So you see the observed species actually remain very, very low. So actually, if you take antibiotics, right, try not to take probiotics too soon. Otherwise, you'll suppress the normal growth of your bacteria. And yet you don't want your bacteria to be just Yakult. You want them to be containing many, many different things, right? Currently, there's so much we do not understand about the gut microbiome and how it influences our health. But we slowly transition into consuming more alternative proteins. Whether for climate or personal reasons, it's very important that we also consider the effects of how these new proteins can alter the gut microbiome, leading to potential changes in health. So in conclusion, our health depends heavily on what foods we eat. And obviously, the foods we eat is determined by our climate, what our climate can support. And with a change in climate and environment, we have to adapt by producing alternative proteins. But the determination of the short and long-term safety of such foods are important. And of course, while we consider the macro environment, which is climate change, we also need to be mindful of whatever goes on in our body or the micro environment. The micro environment within the body is actually very complex, more complex than we think and understand. So new research have shown that there are many, they can affect many different kinds of diseases, and we really have no idea what the mechanisms are. Hence, it's important to be mindful about how we alter these when we are designing future foods, which we intend not just to take as a supplement, but to eventually replace what we are eating right now. Okay, and this comes to the end of the talk, but before I end off, I want to quickly introduce to you, since you are education professionals and also students, a non-profit organization called the Association of Official Analytical Chemists, or AOAC. So a little bit about the history of AOAC. AOAC was originally, it's not called analytical chemists, but agriculture chemists, due to its roots during the 1800s. It was formed together with the USDA, US Department of Agriculture, and later part of FDA, but later become independent in the 1970s. So right now what they are doing is that they are advocating for harmonization of methods, and also in terms of food safety and also public health. And right now it's called the Association of the Analytical Collaboration. For AOACC, because US has their own concerns, US is as a country itself has their own concerns. We in Southeast Asia have our own unique concerns, like we use more herbal medicine, and then our food has more spices and so on, which is not very concerning of the USA counterparts. So we in Southeast Asia, consisting of the ASEAN countries, all came together to create what we call the Southeast Asia section, so that we can advocate for our own rights, advocate for our own concerns. In the AOAC Southeast Asia sections, there are 5 working groups: the capacity building working group to develop competencies and capacities, the emerging issues working group to monitor emerging issues in our region, the harmonization of methods working group to identify methods to harmonize them within Southeast Asia to enhance trades and so on, the microbiological working group to implement microbiological food safety, and lastly, which I want to describe to you, the training of young scientists working group. And this is to establish programs across Southeast Asia to develop, train, and encourage, support, and recognize young scientists like you guys. Annually, we have conferences, meetings. The first meeting was held in Singapore under the theme of food safety, regulation, and analytical science, where we look at things like the regulation, the analytical methods, and so on and so forth. The second meeting was held this year in Vietnam, and the theme was facilitating compliance for food safety and quality in international trade. Some of the topics covered includes method standardization to support cross-border trade, analytical methods, what are the challenges and solutions, and also we have things like technical workshops. And very importantly for students here, okay, we have student awards programs. And for this year, for 2022, oh, there's a question. Please fill this form before signing off. Okay, I'll do that. So we have the Students Award Program, and then we flew students to Vietnam. Okay, we invite students to submit abstracts. Then you fly 12 winners to Vietnam. Okay, we give them $200 travel support, and then from there they will present the posters, and then from the 12 winners, we selected 3 best posters, and we gave prizes of 500 Sing dollars each. Quite a good prize. So we attracted 7 countries and many different schools that participated. So that all these students won fantastic prizes, and they gained a lot of experience presenting and also interacting with the experts worldwide. The conferences that we hold, we invite worldwide experts from US, from China, from Southeast Asia, India, and so on to participate. So that the students really had a very rich interaction. And also, lastly, other than the yearly student award programs, we also have training programs, and this is happening tomorrow. This is a research and industrial training talk, where the different speakers from Singapore and Indonesia and Malaysia will share their experience on what they look for and how they train intern students. So do look out for this. If you come and answer correct questions, you will stand to win prizes at the end, and also you will be given a certificate after participation. So do take part Yeah, so this comes to the end of my talk. Thank you very much.
Thank you, Doctor. If anyone have a questions, any questions, you may type in the chat box and I will read it out for you.
Yeah, feel free to type it in the chat box and also to ask personally.
Yeah. So, questions?
Hi, Dr. Cheng.
Yes.
I'm Aula from Faculty of Medicine. So that was a very interesting talk. So I have a few questions.
Yes.
One, have you tried yourself eating this cultured meat? Have you ever tried it? Or okay, that's the first question. Second is, since you've given the good and bad side of it, the advantages and disadvantages of it, so is it advisable to eat this cultured meat because it doesn't have this? It alters the microbiome, and it's an alternative, but it can alter the microbiome. So is it advisable? And thirdly, how is because it's already sold in Singapore. It's already being sold. So how is the response from Singaporeans?
Right.
Thank you.
So let me answer the first question. Have I eaten cultured meats? So right now I want to clarify, it's cultured meats and not the plant-based meat. Okay, plant-based meat is very common. It can be sold anywhere because there are not a lot of regulatory concerns. Essentially, it's just plant, soy protein. It's the cultured meat that's the problem. Yes, I have eaten cultured meat before. Because I'm in the regulatory committee, when there's an opening and so on, we were given some meat. So I was like, this company is Eat Just. Eat Just has been is given license to legally sell its meat in Singapore. Eat Just is a cultivated chicken meat. So what I was given was two kinds. One was like a chicken nugget, and the chicken nugget actually tastes like fish cake because it doesn't contain the structure of the meat. So they are all cells compounded together, and then the chicken nugget, and the chicken nugget really tastes like fish cake. Right now, a lot of research are trying to see whether they can create scaffolds so that the cells are grown on the scaffold so that they resemble meat better, the texture. Also, they give me satay. They extrude the cells, they make it into like threads, and they put it on the stick, and then they dip it into the satay sauce. But of course, everything you dip into satay sauce tastes like satay lah. So it's nice, it's good. Second, would I say it's safe to eat this kind of meat? It is a very difficult question. After we have gone through the regulatory process, we will determine that this is safe to eat. But right now, what we understand is these meats are eaten as a side. The daily intake is not a lot. We really need to do tests on larger intake, larger consumption. Then we'll know what is the long-term effect. So right now, it is still something that we want to see how to understand some more. Yeah. And what's the last question? The third question?
How is the acceptance in Singapore towards this meat?
Acceptance in Singapore is I wouldn't say it's fantastic so far. The reason is because these are very difficult to create. So they only create small batches. So it's not like sold in supermarket. Anybody can buy. It's only given to say certain high-end restaurants to create like creative foods for consumption. So there's no market data on this yet. But the plant-based meat is quite popular. Yeah, I'm not too sure whether you have tried Subway. Subway actually use the Impossible meats. It's a plant-based meat. They try to mimic beef. So this is widely accepted. Yeah.
Thank you.
Thank you. So that's it.
This is not a question, but for me, there's also the issue of halal when we want to eat.
Yeah, correct.
So,
So yesterday I was in the regulators forum. Saudi Arabia was talking about halal issue. So they are still debating on this. Basically, what they think is that if the cells are taken from animal that's prepared in a halal way, then basically it can be considered halal. But of course, this differs from country to country. Still trying to debate whether the cells should be from a live animal or should be from a slaughtered animal in a halal process. So these things are still being regulated and discussed amongst the scholars, the Islamic scholars. Yeah, thank you for your question. This is a very good question. Okay, there's something in the chat box. Q&A session. Feel free to ask any questions. Yes. Feel free to ask any questions. Okay. Any other questions? We are at it's 12. Slightly past 12. Are there any other questions that you would like to ask? You can ask them here, or you can also email me. I'll be happy to answer over email also. No, oh yeah. So if you would like to sign up for membership, it's a free membership for students right now at AOACC website. You can just Google AOACSEA, and then you will be able to find the website to sign up for your free membership. Yeah, right now it's still free. Might be considering having fees in future, but for students right now it's free. Yeah. All right.
We close our session of the day.
Okay. Am I supposed to fill in the form also?
No.
No. Okay.
It's for the attendees. Well, with that, I would like to end our session for today. Thank you, Doctor, for the interesting and informative presentations. And thank you for all for joining us for today.
Thank you very much. See you. Bye.
thank you.